So in this video this will be the concluding part of genetic code, the final part. We will look at what NCERT is trying to say about the deciphering of the genetic code. So if you have read your NCERT, then it says something in this particular paragraph, something about Marshall-Nirenberg cell-free system, Severo-Ogawa enzyme, polynucleotide phosphorylase, the enzymatic synthesis of RNA. These are the terms, the homopolymers, the copolymers. So understanding NCERT requires something, some basic concepts to be explained in detail, and I will try to make a sense out of all these things which are given in your NCERT textbook. Normally what happens, that student actually just just cram up these particular points which are written in bold and just think that it is over. But look at the NCERT. I have already discussed something about the complementarity, etc. Now we will be looking at this last paragraph, this paragraph on page number 112, that what exactly NCERT is trying to see. Now we know that genetic code is a triplet. There is a codon. This codon is made up of 3 letters. There are 61 codons that actually code for 20 amino acid. We know this. So we know about degeneracy, we know about unambiguity, we know how to read an open reading frame, we know the dual role of AUG, the universality of the genetic code apart from it being a triplet code. So this is what we know. Now my idea is that what I want to do is that how do we actually came to know that which codon out of these 61 codes for which amino acid? How was it possible? So in the beginning of the video, I will take time and will tell you the 2 important things that I have shown you in your NCERT textbook in the beginning itself. One is, your NCERT says that Marshall-Nirenberg's cell-free system. So this is what we are trying to understand first. See, if you want to score very well and if you want to be very successful in your life, do not just cram up everything. Try to understand. The purpose of the science is not cramming up. You must be able to understand what exactly is written. So let me tell you the thing that I am going to tell you this time. So they are saying that there was a cell-free system, Marshall-Nirenberg's cell-free system for protein synthesis. So if we can show it clearly, so this is the simple word that we are trying to now explain. What exactly is a cell-free system? So you know that there are large number of chemical reactions occurring within the confines of a cell. So your NCERT says in your class 11th NCERT, the chapter 1, that cellular organization with metabolism is a defining property of life. So there are so many reactions occurring in the cell, but these are not isolated reactions from one another. So cell is a very, very complex thing. These things are actually interacting with each other, influencing each other. So that means there is a huge amount of complex interaction between the various components of the cell. So when we say that something is happening, there are let us say hundreds of reactions occurring within the confine of this particular cell, then what I am trying to say is that each and every one has a potential of influencing or modifying the other process. So they are not independent. Now therefore, to understand what is happening in a particular reaction within the cell will be a very difficult proposition. So I hope this thing is absolutely clear. So if I am trying to understand a particular thing within the cell and I want to understand one particular point or one particular reaction that is occurring in the cell, it will be a very, very difficult task, because I know that in this cell there are huge number of chemical reactions occurring together and each has a capacity of influencing the other. So this is very important. So I require what is called as a cell-free system. So the word is self-explanatory. So that means it is what I am using is I am using the components in isolation. So what I will try to do, I will try to the components of the cell, I will try to isolate them. So that means now they are not present within a cell, and then I will understand the individual components. This can be done by centrifugation. So this is what is called as a cell-free system. So in a cell-free system, you are actually making it easy. to understand one particular thing so that it is not influenced by all other things that would be possible in that will happen in an environment of a cell. So now we understand what exactly cell-free. This is a literal thing. So that means I have extracted the components of the cell, and then I am individually understanding what exactly is happening within a component of a cell. So that would be a cell-free system. So this cell-free system has a very, very important role to play even in biotechnology and in molecular genetics and basically understanding the molecular biology. So again, I am trying to emphasize that it would not be possible to just understand the thing completely when it is occurring inside the cell. So I have created a cell-free system, and this is the cell-free system that NCERT is talking about, that the cell-free system of Marshall, Nirenberg and Mathai. There were two scientists, so Nirenberg and Mathai. So we are looking at two scientists actually, Nirenberg, and we are looking at the experiment by Nirenberg and Mathai. This is double T. So that means these two scientists, obviously we are giving credit to Nirenberg for cell-free system, but please understand that deciphering of the genetic code was contributed to by many scientists, and you know that a very famous Indian scientist Hargobind Khurana was also pivotal in our understanding of the basics of the genetic code. So primarily we are talking about the experiment by Nirenberg and Mathai, and Nirenberg's cell-free system. You have understood what exactly is a cell-free system. So this cell-free system can actually be created in two methods. So there are two ways by which you can actually make a cell-free system. One, I have already told you that you extract the components. So this is an extraction based cell-free system. This was the system that was actually used by Nirenberg in his experiments. There is another mechanism by which you can make it an enzyme based cell-free system. So that means now we are not looking at a particular component, we are looking at a biomolecule, we are looking at an enzyme. You know, most of the enzymes are protein and they will be catalyzing the reaction, and I want to understand the reaction in complete detail and precisely. So I have created an enzyme based cell-free system, and like Nirenberg and Mathai, I can actually create what is called as an cell extract based cell-free system as well. So I hope now we understand what my NCERT is trying to say. So this is very, so many things that are occurring in the cell now can be performed in a cell-free system in isolation. This can be done. So this is what is called as cell-free system. This is very, very simple. One more thing to discuss in the beginning, then we will come to the actual deciphering of the genetic code. First we understand the basic terms, what is happening, what my NCERT is actually talking about. And let me show you another word. And this another word is Severo-Ogawa enzyme, what is called as polynucleotide phosphorylase. So let us look at this enzyme in slight detail. So let us understand what exactly is meant, what exactly this particular paragraph of NCERT is saying. So now we are saying that okay, there was an enzyme, and this enzyme is called as polynucleotide phosphorylase. So this is a very, very important enzyme, and it helped in understanding what codes for what in the genetic code. And now understand this, that what this enzyme is. So this enzyme was discovered by a scientist. The name of the scientist is Manago and something like that. I am not absolutely sure about the spelling, has a weird spelling, and he was working in the lab of a scientist who was called as Severo-Ogawa. So this person was the boss. He had a lab, and this other scientist, please check the relevant spelling of the name of the scientist. They actually discovered this enzyme, what is called as polynucleotide phosphorylase, and therefore it is sometimes called as Severo-Ogawa enzyme. But let us not forget the name of this scientist. I have already forgotten the spelling of this particular name. So please give credit to this scientist as well and just try to find out what exactly is the correct spelling of this. You can easily understand that how we actually conveniently forget the contribution of many great scientists. So this is interesting, and please make note that you will look at the internet for this particular thing. So now pay attention to the big boss, which is Severo Okawa, the person who had lab and actually who shared the Nobel Prize with Nirenberg and Mathai for the contribution towards understanding or deciphering of the genetic code. Now when this enzyme was synthesized, was isolated or was identified, what actually it does. So the first activity is a 3' to 5' exonuclease activity. Now try to understand, it is an exonuclease. Nuclease is an enzyme that will cleave the nucleic acids or will digest the nucleic acid, and exo would be from the end. So that means if this is a chain, this is a chain, and this chain is 5' to 3', so this direction would be 3' to 5', and it can cut the nucleotides from the end one by one. It will not cut directly in the middle or directly in between, because it is not an endonuclease, it is an exonuclease. So this is the primary activity. But strangely it was shown that it could actually synthesize RNA molecules. So it could actually synthesize RNA molecules. That was very surprising, which means that we know that RNA molecules are actually made up of ribonucleotides. So it could use ribonucleoside triphosphates. You know what we are saying. You have already heard this word about in transcription as well as DNA replication. What is a nucleoside triphosphate? And it is very similar to ATP. If you don't remember, let me remind you again. And again, if you are having some problem, just go and see the video on DNA replication, and then you will understand that what is the similarity between nucleoside triphosphate and the ATP molecule. And these ribonucleoside triphosphate will serve the dual purpose. They will provide the energy for polymerization and as well as will act as the substrate. So I take it for granted now that you have seen the initial videos and you are comfortable with what I am saying. So that means they could pick up ribonucleoside triphosphate and can actually attach them in vitro in cell-free system. This is very, very interesting, and very perplexing was that it did not require a template of DNA for that. So template was not required. Now let us be absolutely clear. This is the main function of polynucleotide phosphorylase. It is actually an exonuclease, but this is an additional property this enzyme has, and this enzyme can enzymatically synthesize a sequence of RNA, a molecule of RNA, by joining ribonucleoside triphosphate and using them as source of energy and cleaving the phosphate groups from them and joining the ribonucleotides. And very interesting thing was that it did not require a template of DNA. We know that actually RNA synthesis occurs on the template of DNA. We have seen in transcription that all major cellular RNAs are actually synthesized on the template of DNA, but they found an enzyme which was actually capable of joining the ribonucleotides without the need of a template of DNA. That means you could now enzymatically synthesize RNA molecule in a predetermined sequence. So you could choose the nucleotides that you want to incorporate into a RNA molecule. So this was possible with the discovery of polynucleotide phosphorylase, otherwise also known as Severo-Okawa enzyme. So let us come back to NCERT. Let me show you what NCERT is saying. So the chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combination of bases. Marshall-Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severo-Ogawa enzyme, polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in a template-independent manner, which is enzymatic synthesis of RNA. This is a very, very important line of NCERT. I hope now you have understood what your textbook is saying. Now let us see what Nirenberg did. So what Nirenberg did, that he enzymatically synthesized an RNA, and this RNA actually contained poly-U. Now please understand, so we are just making a homopolymer. This is what your NCERT is saying. So I am actually joining only uracil, only the ribonucleotides that contain the nitrogenous base uracil. Now this is basically RNA molecule, and this RNA molecule, can you see, is a homopolymer. So these are the repeating subunits, these are the monomers, and because all of them are absolutely identical, we call such a polymer as a homopolymer. This we have discussed in your biomolecules chapter in class 11th as well. So it is very important. Let me remind you again, if you are reading the molecular basis of inheritance, you should have read the biomolecules chapter well first. That is how you go about it. If you are reading the Mendelian genetics, please first see the chapter on cell division, then it will become very, very easy. You cannot keep them separate. So one is the building block of the other. So without a strong foundation, you cannot make a strong building. So a weak foundation cannot have a strong building. So this is very important. Now we understand that this is actually what Nirenberg did. And when he got this particular, please understand, now we have a cell-free system. In a cell-free system, there would be a ribosome. The ribosome is capable of protein synthesis, and it actually translates a molecule of RNA. And I have provided the ribosome with this RNA molecule, which is homopolymer. You know that the genetic code is actually triplet. So this would be, you know how to read it. These will be the codons. And as you can see that every codon is U. So every codon is this. And when this particular RNA will be translated by the ribosome, it will be very interesting to see that what is the protein formed, because you know that because each and every codon is identical, so that protein should also be a homopolymer. So naturally occurring proteins are never homopolymers. Naturally occurring proteins are never homopolymers. This is what we are deliberately doing in a cell-free system. In a lab, we are enzymatic synthesis is being involved here. So what they found, that the peptide that was actually translated by the cell-free system actually contained only phenylalanine. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So it contained only phenylalanine. So this PHE means that it is actually phenylalanine. It was actually phenylalanine. So this phenylalanine, now we know, must be coded for by UUU. This was the first codon that was deciphered by Marshall-Nirenberg. I hope this thing now makes absolute sense. So this is now very easy. So that means now you have joined a homopolymer. So let me show you the NCERT again. So what NCERT is saying is something like this. So it is saying that well-defined combination, homopolymers and copolymers. Can you imagine? So this is the word that we are trying to explain in your NCERT textbook, that what exactly would be homopolymer. Now, after the first codon was deciphered, after we came to know that okay, UUU codes for phenylalanine, there was a race to decipher all other codons, and there was a race between two teams, a team led by Severo-Ogawa and a team led by Nirenberg. So they were trying to outcompete each other, and that actually became a very healthy competition for people like us who are students of molecular biology, because they did very pioneering work to actually decipher the genetic code, because once the method was known, then everything fell in place very easily. So it can be very, very easily understood now that I can actually make another homopolymer, and that would contain the ribonucleotides, say for example only say for example C, cytosine. So I am making a poly C. So whatever peptide, the peptide, whatever amino acid will be present in it would have the codon as CCC, and it turned out to be proline. So this was, I am again telling you a word of your NCERT, the homopolymers. So again, trying to remember the codons. It will be prudent if in the initial stage you remember what are the homopolymers codons that code for which amino acid. So we know one which is UUU, this codes for phenylalanine. We know CCC, that codes for proline. And if you add only A, so you enzymatically synthesize a RNA molecule that has only A and then get it translated. So the amino acid that will be present will have the code AAA, and we will see again a homopolymer, and this time it is lysine. At a very later stage, it was understood that actually GGG codes for glycine. So GGG codes for glycine. This was the step number 1. So step number 1 was you make a homopolymer RNA and get it translated and see the peptide. What exactly is the amino acid? All the amino acids should be identical. It should be a homopolymer. And immediately you know, at least for these 4, that what will be the codon for which amino acid. This is what your NCERT is trying to say. And the second term that I will actually show you is, so this is homopolymer that we have understood. Now we are looking at what exactly is meant by copolymers. So that means now instead of taking only one nucleotide, let us take a combination of 2. So let us take a combination of 2. Now this is the second step. This will be very interesting. Please try to understand what I am trying to say. This would be for let us say AIMS entrance exam, or for a very, very good rank in your NEET entrance exam, as well as for your general understanding and confidence that you know exactly what NCERT is saying, what this copolymer is. So that means now when I am enzymatically synthesizing RNA, I am providing the polynucleotide phosphorylase something as a substrate, and that would be ribonucleoside triphosphate. So I am taking 2. So in the medium, I am giving this ribonucleoside triphosphate and this ribonucleoside triphosphate. And what I have done that I have taken different concentrations in the medium. So this is 5/6, this is 1/6. Now please understand what I am doing. I am giving my enzyme in the environment these 2 types of, so I am now trying to make what is called as copolymers, not homopolymers. But what I have done, that I have deliberately kept the concentration of one of them as 5 times higher than the other. So this is very, very interesting. You know the randomness. So you know that this getting incorporated has a 5 times higher probability than C getting incorporated into the chain. So we know that the different RNA molecules that will be formed, they will have, some of the codons will be A on the one hand, and obviously all 3 can be C as well. And some of them will have 2A, 1C, and some of them will have 2 or 1A and 2C. This is the combination that is possible. So you know that they will be getting incorporated into the RNA molecule, and we know that this enzyme is now picking up only A and C. and therefore there will be some codons which will be AAA, there will be some codons which will be CCC, and there will be some which will be 2A 1C and some which will be 1A 2C. I hope this is being understood and followed by you. Now this is very easy. Now because the concentration of A is much higher than that of C, therefore the concentration of this codon in the RNA molecule will be the highest. This is very, very simple. So if you are adding from a solution or from a group, from something that is being provided, it is a combination of A and C, and you are putting one and you are picking one and putting it here. So therefore if it is A, the probability will be 5/6. For the simple reason that the amount or the concentration of A is 5 times that of C, and we know from our earlier discussions that individual events occurring simultaneously, the combined probability is a product of their individual occurrence probability. So at the first point I have added A, the probability was 5/6, and at the second one again I am adding what is called as A, so again the probability will be 5/6, and same for the third. So which means that this probability combined, the probability of combined, so if there were 216 codons, then this would 125 would be AAA. Now this makes sense. This is simple mathematics. What about this? This would be 1/6 into 1/6 into 1/6 into 1/6. This would be 1/216. So immediately you can see that what I am trying to do, I am simply using mathematics, the probability. I know the randomness of the enzyme, and this is how they will be oriented. What about 2A and 1C? So this would be 5/6 into 5/6 into 1/6, or 25/216. And what about this one? This one would be 5/6 into 1/6 into 1/6, or 5/216. So I know that this would be 120, this would be in the highest concentration, this would be 125/216, and triple C would be 1/216, and I already have deciphered that this codes for lysine and this codes for proline. So I know in the mixture I will get this much, what? This much would be which amino acid? Lysine, and this one would be proline. This is absolutely sure. And now if I make some logical conclusion, any amino acid which is actually 5 times than that of proline would have 1A 2C. So in the mixture, an amino acid that is found in a concentration that is approximately 5 times higher than that of proline would be having 1A and 2C. This is very interesting now. And similarly, anything which is around 5 times lower than lysine, then that would be containing 2A1C. So now scientists could understand that the amino acids, there will be some of the amino acids that will be in this concentration, they should contain 1A 2C, and the others will contain 2A1C. So this is a copolymer that I have made with the help of A and C. You can repeat the experiment with A and G. You can repeat the experiment with C and G. You can repeat the experiment with U and A. Then you can understand so on and so forth, that for almost all amino acid you will at least know that what is the number of which letter that is present in them. So I hope this is absolutely getting clear to you. So this was the second step of deciphering. This is what NCERT is meaning by copolymers. So this was the second step. Now we know that this particular amino acid that is about 1/5 of lysine in the mixture will have 2A 1C, and similarly something which is 5 times more than proline would be having 1A and 2C. So this thing is now getting absolutely clear to me. Now there was a third step as well. So this is where your NCERT actually stops, and I will just give you an idea of the final step of deciphering of the genetic code. I will not go into the unnecessary detail, but just to complete the picture. Now suppose you know that the third step would be that an amino acid has 2A and 1C. Now you want to know whether it is AAC or it is ACA or it is CAA. Can you imagine? These can be the codes for an amino acid that has 2A and 1C. So this was the last of the component of the jigsaw puzzle. And later on, when you go into your higher classes, there is a very important enzyme which is called as Aminoacyl-Trna Synthetase. This enzyme will help us understand which exactly codes for what, and that led to the deciphering of all the codons of the genetic code or the mRNA codons on the genetic code table. So this is a very, very interesting thing. As I told you, a Nobel Prize was shared by Marshall Nirenberg, Nitin Mathai and Severo Okawa. Now I hope you understand your NCERT well. Now please pay attention to each and every line of NCERT. I have tried to explain the NCERT and every word to you. Now it should make sense. Do not just try to cram it up. If you understand something, then the dependence on cramming is reduced, and actually it becomes more fun and you become more and more confident. And again I am telling you, just logically thinking and reasoning, apply reasoning to that table that is given of messenger RNA codons in your NCERT textbook. Incidentally, without looking at, I probably know the page number as well. It is 112 of class 12th NCERT. Correct me if I am wrong in the comment section. And then please understand that you will, when you understand the third code degeneracy, when I have told you that so many of them are actually are made up of are coded by 6 codons, then it is very easy to understand. Only by remembering 14, 15, 16 codons, I can actually know what are the codons. Say for around 40 codons they code for which amino acid. And now I am ready for the exam. So either the question will be solved by inclusion or by exclusion. So this is very important. So right at the first time, right at the first moment, just try to understand what I am trying to say. You know what is AUG. There is one codon that you know. So AUG will be methionine. You know this. Similarly, immediately comes to the mind is but a UGG. Now UGG, you know, is coding for tryptophan. These are the 2 amino acids that are coded for by a single codon. So immediately this is in my mind. And then you know about the 3 stop codons, UAA, UAG, UGA. So you know about this. So this makes 5. And then you know about glycine. And you know glycine GGG. So you know the homopolymers. And then just see that which of these homopolymers actually show the third code degeneracy. So if you know GGG, you know 3 as well. So this is how you pick up, and this is how you apply the knowledge to that particular thing that is given in NCERT, and you will see that just 40 minutes of careful observation of the table will give you a confidence of predicting the sequence of virtually every amino acid, either by inclusion or exclusion. So I hope this very, very important thing, the genetic code, is now absolutely clear. So now we know everything before translation. We know what is the genetic code to be translated. We know which is the adapter molecule, the transfer RNA molecule. We also know about the machinery that is going to synthesize protein, which are the ribosome. So that means after understanding all these 3 things, now I am in a position to very easily understand the actual process of translation, that is formation of protein from an information that is given in the messenger RNA molecule. Remember, it will be done at the ribosome. So in the next videos, we will look at the process of the translation, the final step of the central dogma of molecular biology.